Good morning. For our participant who's already in this meeting, please fill in the attendance form that given by the operator in the chat box.
Hi, Dr. Tian.
Hello.
Can you hear me? Hi.
Yeah. How are you?
Good, very good. How are you?
Good,
We
good.
just wait for some introductions.
Okay. Yeah. Wait. No worries. Okay. Okay.
Okay, good morning, Mr. Jonathan and Mrs. Ratna.
Yeah. Okay. The host.
Okay. Mrs. Ratna, can you hear me?
Yes, yes, I can hear you.
Okay. We will start our meeting today. Assalamualaikum warahmatullahi wabarakatuh. Excellency, Professor Dr. Widowati, SSE, MSE, the Dean of Mathematics and Science Faculty of Diponegoro University, Honorable Dr. Susilo Haryanto, SSE, MSE, Head of Mathematics Department, respected all of lecturer and teaching staff of Mathematics Department, to the Honorable our speaker, Mr. Tian Hai Tian, Associate Professor at School of Mathematics, Monash University, Melbourne, Australia, and to our moderator, Mrs. Ratna Herdiana, MSc, PhD, unforgettable one, whom I love. First of all, let's pray and thank unto our God, Allah Subhanahu wa Ta'ala, who has been giving us mercies and blessing, so we can attend and gather a meeting in this place in good condition and happy situation. Standing in front of you all, my name is Mega Permatasari. You can call me Mega. I am honored to be your master of ceremony on this special event, visiting lecture with stochastic modeling and simulation of genetic regulatory network as the topic. As your master of ceremony, I would like to read our agenda in this meeting one by one. The first agenda is opening. The second agenda is lecture and Q&A session. And the last agenda is closing. Ladies and gentlemen, before we enjoy our meeting today, let's open our program by pray together. Pray begin.
Okay. Yeah.
Finish. Thank you very much.
Thank you.
Okay, ladies and gentlemen, now we will move to our main agenda. But before we going to our main agenda, I will welcome all participants to turn on their camera and be ready for photo session. I will give you 1 minute to get ready.
Okay. Okay. So, what I should do?
Okay, I think no one will turn on their camera, so we can start our photo session.
Okay.
We will start for the first slide. Okay.
Oh, yeah, okay. So sorry for that. I didn't realize that's my.
Just a moment, Dr. Tian. We will
Yeah.
take pictures first for documentation.
Okay. So first take a picture.
Yes, yes.
Yes.
So I need to stop sharing.
Yes, stop sharing first.
Okay.
Okay.
Thank you.
Okay, we will start our photo session for the first slide. 3, 2, 1. Next slide. 3, 2, 1. And last slide. 3, 2, 1. Okay, thank you very much. All participants will turn on their camera. Ladies and gentlemen, now we will going to move to our main agenda, which is lecture by Dr. Tian Haitian, who will guide by Mrs. Ratna Heriana, MSc PhD. Before that, I would like to introduce Mrs. Ratna as our moderator. Mrs. Ratna Heriana, MSc PhD, is a lecturer in Mathematics Department, Science and Mathematics Faculty of Universitas Diponegoro. She finished mathematics undergraduate in 1988 from Institut Teknologi Bandung and a whole double degree in mathematics from the University of South Wales in 1999 and a whole PhD in mathematics also from the University of Queensland in 2003. She has written about the optimal control of smoking cessation program for 2 subclasses of smoker in 2023. Okay, without any further ado, please welcome Mrs. Ratna, MSc PhD. Mrs. Ratna, can you hear me?
Yes.
Okay, time is yours, Mrs. Ratna.
Is my voice clear?
Pretty clear.
Okay, thank you, Ms. Mega Parmana. You've done a good job as MC so far. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. A very good morning, ladies and gentlemen. To respected Dean, Vice Dean, Head of Departments, and the Secretary, my fellow colleagues, also the students and all participants in this visiting lecture program, I welcome you all and thank you for joining in this program. And of course, very warm welcome to our guest lecturer for today, Honorable Associate Professor Dr. Tian Haitian from Monash University, Melbourne, Australia. Hi, Tian. Good day, because it's already noon, right there?
Yeah.
Yeah, it's still here morning.
Yeah. It's noon now. Yeah.
Yeah. It's an honor for me to be the moderator for this morning session lecture. Dr. Tian is my office mate when a very long time ago when we were studying in Queensland. And I know he's very clever. So I used to ask him if I don't understand something. Okay, let's go through his brief biography.
Yeah.
Okay. So his position is Associate Professor at the School of Mathematical Science, Monash University, Melbourne, Australia. You can see his contact and check his website for more details. His education background, he did his PhD in mathematics at the University of Queensland in Australia, graduated 2001. He did his master's degree in applied mathematics, completed 1988 at Huazhong University of Science and Technology, and his BSc in computational mathematics, 1982, also from the same university, Huazhong University of Science and Technology. His research interest is on stochastic modeling of genetic regulatory networks. Yeah. A mathematical modeling of cell signaling pathways, mathematical modeling of cancer initiation and progression, stochastic simulation of chemical reaction systems, and also Bayesian inference for estimating parameters in biological models. His employment history from 2011 until now, he is the Associate Professor at the School of Mathematics, Monash University. Prior to that, he was a reader and Lord Kelvin Fellow at the School of Mathematics and Statistics, University of Glasgow, United Kingdom. Yeah. Academic awards. Yeah, he some achievements, future fellowship from the Australian Research Council, from the University of Glasgow as Lord Kelvin Fellowship, and also from Hubei Province Government, China, this Excellent Research Award. Okay. He has also been successful in obtaining competitive research grants. So you can see the list here from Australian Government Research Council, from also from UK, United Kingdom. Yeah, so he has many research grants that he has obtained. Yeah. So it's very productive. Some of his selected publications. Oh yeah, here you can see the publication from last year, 2022. He published how many? 10 articles, paper in reputable journals. So it's very productive. And he's enjoy in his as a mathematician, I think. Okay, with that. Oh, by the way, Monash University, is ranked 44, is it? Top 50 university in the world. Yeah. I think we have a branch in Jakarta, but
Yes.
In business, I think, Monash University. You can read or see the publication. Is more on lately. Is more on the gene regulatory networks. Yeah.
Yeah.
So the topic for today, it's related to that, how we model using stochastic modeling of the gene regulatory networks. So I think that is his brief CV. Thank you. So yeah, Dr. Tian, we have 2 to 2.5 hours of your lecture. Yeah, because unfortunately the dean and vice dean has other agenda, cannot join us today. We are busy with accreditation this week. So to the students or participants, please take notes. And I think this is very interesting topic that Dr. Tian will deliver. So pay good attention and ask questions. If you would like to write in Indonesia, that's no problem. I can just read it in English. Write it in the chat room or raise your hand during the question and answer session. So, Dr. Tian, you ready?
Yeah.
Yeah, okay. I think we can start the lecture, please.
So I share the screen.
Yes.
Okay.
Yes, it's clear.
Okay, so first I thank the Dean for the opening ceremony and also for introducing me. First, I thank Radna for the kind invitation. So I have this opportunity to discuss very interesting problem with you today. So today I will discuss stochastic modeling and simulation of genetic regulatory network. So this is all the materials here. So if we have time, we will finish all the slides. If we don't have, so we may finish at section 6. Okay. So this is picture for the mathematical modeling. So if we studied the mathematics before, we normally from step E. So normally we have a differential equation, and then we will study the property of the differential equation, and may also get a simulation of the system, like the step F here. But if we consider realistic problems, like biological systems in finger A, so suddenly there will be some cells or even tissues, so the structure would be very difficult. So in that case, we may have to formulate a model system. This model system is step B. So here it is still a biological system, but the structure of the system is much simplified. So in that case, for example, we may have two variables, two molecules in the system. We use red and blue to represent that. And then next step, we need to find the relationship. So what is so-called the regulation between these two molecules? So this is step C. So in that case, we may have certain, activation, degradation, or some, chemical reactions. So this step is understanding of biological system. We need to find the regulation in the system. And then from step C to step D, I think this is the step for stochastic modeling. So in that case, we need to find what's the chemical reactions in the system. So for example, so how to from, to generate molecule X, what is the degradation of X, and so on. In that case, we need to find the process of these reactions in the system. This is the first step for the modeling. In the second step, we need to from the chemical reactions to get the differential equation. So with differential equation, we can consider, what is the formulas inside that, so we can analyze the property of the system. So as I just mentioned, step E is the, for the start of mathematical analysis, but from C to D is the start point of the stochastic modeling. So we need to find what's the reactions there. So today we will first discuss some basic principles how to formulate the chemical reactions in gene regulation. And then we don't go to step E, because for step E, it is a deterministic modeling. So we will consider direct simulation of the system in step D. So this is the major topics in this discussion. Okay, so if we consider the concept for mathematical modeling is to use mathematical formulas to describe real world problems. Here, certainly we need to consider certain assumptions because real world problem normally is very complicated. We have to study a relatively simple system, and then we can use mathematical equations for that. So for mathematical modeling, we have two purposes. The first one is to understand the mechanisms in the biological systems. So sometimes experimental study may have certain observation, but what is the mechanism inside that? So biologists may not be know. So in that case, we may use a mathematical model to find the underlying mechanism. Another way is to make a prediction. for example, what's the population of ecological systems, what's the case for cancer disease, after 5 years treatment or so on. So that's for the prediction. For mathematical modeling, I just mentioned that we need a certain assumption. This assumption is critical for the quality of mathematics. Because if we consider any differential equation, if we have certain unknown parameters, we can always estimate the parameter and then let the equation to match experiment data. If the assumption is not correct, so sometimes we may we can't write a good paper for mathematical modeling. So in that case, assumption is critical, for mathematical modeling. So this is the general step. So when we consider mathematical modeling, I hope to say, actually, when we consider modeling, we already have the purpose of the model in our mind. What is the possible result we hope to realize? So this is the starting point for the problems and also for all the formalisms in the process. So generally, if we have problems, we can make an assumption and then get the formula, find the solution, and then find that solution would be the prediction. We may also, from mathematical analysis to get some theoretical result. But generally, so we use differential equation or other mathematical formulas to make the numerical simulation. So if the prediction is okay, so the model is fine. If the prediction is not good, so it's entirely different from our expectation, we may change the assumption, do the modeling again. So next, we will consider genetic network. Genetic network is for gene expression is the process to convert the information in the DNA into the functional molecules. Here we have two types of functional molecules. That's the messenger RNA. So that's the directly from DNA. And then we also consider protein. Protein is the product from messenger RNA. So in gene expression, we have two steps. The first one is transcription. That's the step from DNA to synthesis messenger RNA. The next step is for translation. That's from messenger RNA to synthesis protein. So here is the picture for the central dogma. You can see here we have two steps. That's from DNA to from transcription to get RNA, and then from RNA by translation to get protein. The steps is here. So this is a picture for a cell. This is a nucleus. DNA is inside the nucleus. So initially, DNA is a closed form. So in that case, we can't get RNA. So DNA should first transform from the closed form to the open form. And then transcriptional factors can bind into the starting point and then start the transcription. So after the messenger RNA is synthesized in nucleus, it will move from nucleus to cytosol. And then in cytosol, the messenger RNA can synthesize protein. And then protein is the final product to realize the function of DNA. So this picture looks like relatively simple, but actually transcription is quite complicated. For example, this is a more detailed picture, for transcription. So we don't discuss, for the translocation and then from for translation to get the protein. So here I just mentioned that actually when DNA is in the opened form, a number of transcriptional factors are needed to start the transcription. So from here you can see we have a data box. The data box is a region in the upstream of the DNA. So this is the starting point for transcription. And then we have some molecules. This molecule is called transcriptional factor. That's TF here. So we need a number of transcriptional factor proteins to bind to the starting point. And then another molecule is called RNA polymerase 2. So that's the POL here. This is the large molecules here. Actually, RNA polymerase 2 move along the DNA to get the messenger RNA. So I hope to mention, here actually we have a number of transcriptional factors to start the transcription. So this step is give a little bit more detail for the transcription. So for example, there's the empty data box, no molecules bind here. There's the A, and then we have a transcriptional factor, TBF, and then bind to here. And then after that, we have more, transcriptional factors binding together. So in this case, we get more and more molecules here, and then that's the RNA polymerase 2. So RNA polymerase 2 will bind together, and in that case, these molecules should be phosphorylated, and then it can start the process of transcriptional factor. So actually, in genetics, biologists are still studying what is the detailed process for this picture. So here is just a relatively simple picture for that. So we will, based on this picture, to use chemical reactions to consider the gene expression. So if we here we return to the original slides. So the picture here we just get the second step. That's the mental model. So that's, I think we can consider. Okay, this is the mental model for transcription. You can simply consider that, but we have to make some simplification. So here we consider the first step. So if we consider transcription, so normally we don't consider, sometimes depends on how complex in your model. You may consider, DNA plus a TF or DNA plus RNA polymerase. So in that case, we can consider, DNA and RNA polymerase to form a complex. This complex actually is a single variable. And then from this complex, we can get messenger RNA, and then RNA polymerase separate from DNA. So this is the picture for that, because in this picture they use P to represent DNA. They consider this the promoter. So you can see here I just show you when we consider gene expression we may use different notations to represent the system. So here is the case. So this is the first one. We consider transcription to get the messaging array. So we can use M to denote messaging array. We can also use R to denote messaging array. It's the same. And then this step, as we just mentioned, if we consider the previous reactions, here we have two reactions. We can put it together in a single step. It doesn't matter. You can say here. You can understand. Actually, this process represents two reactions. This is the first reaction. This is the second reaction. So in that case, you can simplify. You remove the center, the intermediate step, and then from the start point to the final step. So you can directly, get the one-step reaction. So you can, firstly, you may simplify the process. On the other hand, you can make the process more and more difficult. Because, if you consider this picture, actually, DNA is a non-chain. So when RNA polymerase move along the DNA, actually it has a number of steps. It's not a single step. So in that case, you can consider, okay, they have moved first step, move the second step, and then move the final R step, and then finally you get the RNA. So you can, for this case, you can make the process a little bit more complex and get more reactions. Certainly, if you consider a more detailed system, generally, we can expect this system is more accurate. However, it needs more data to support that. Because in that case, we need to estimate a number of reaction rates for all these reactions. If we consider a simple one, we only need to estimate one parameter. That's enough. So this picture, so give you an idea. So for any biological process, we may use a very simple reaction to represent that. We may also consider many reactions to make the system more detailed and more accurate. Okay, so this is the reactions we have, transcriptional factors. In the previous slides, we don't have transcriptional factor. So in here, you may consider, that is the starting point, and then we have the first transcriptional factor binding to that, and then the second transcriptional factor binding to that, and finally, RNA polymerase binding to that. With all these molecules together, we can, the DNA can synthesize RNA. So in that case, you can consider, we need a number of reactions. There's the binding of first transcriptional factor TF1. There's the binding for TF2. This is the binding for RNA polymerase. And finally, all these complex to get the RNA. So in that case, depending on the requirement, depending on the data, you may use different model to realize the gene expression process. So this is the, in that case, we may, in that case, we can consider, another factor. This is for the repressor. So repressor is molecules, they can't let the transcription begin. So that is if gene is binding with a repressor. So in that case, RNA polymerase can't bind to these molecules. So that means if a repressor binding here, actually it will block the transcription. So in that case, RNA can only bind to protein binding to DNA without a repressor. So that's the case. So repressor is that's the so-called negative regulation. So on the other hand, if we have sigma factor, if sigma factor binding to RNA polymerase, it will accelerate the binding reaction of the second one. So in some sense, sigma factor, you can consider that is a positive regulation. So in that case, for transcription, we may have positive regulation and negative regulation. And then finally, that's for the translation to synthesis protein. So in that case, we have a ribosome bind to the RNA, and then they will move, to get them protein. So this protein is initial product of translation. We needed to post translational modification to let the initial protein to get a functional protein. So that's certainly that's the last reaction would be the reaction in the cell signaling pathway. If we only consider translation, so the first two steps is enough. Okay, so that's the process for gene expression network. So also we have degradation process. So that's degradation, that is RNA will disappear and the protein may also disappear. The mechanism for degradation is complex. For example, if we consider RNA degradation, so that means if I think that's the similar process like our cell. If RNA synthesis one protein, the length RNA will be shorter. And then if the synthesis a number of protein, the length of RNA may not longer enough for further translation. So in that case, RNA will have degradation. So like our human cell, after a certain number of division, the cell wouldn't be functional at that time. Our human will die at that time. It's a similar process. Protein is similar. So in that case, certainly for protein degradation, there is a more complicated process there. But generally, we use only one reaction to represent that. So we may also consider, other molecules involved in the degradation. So, for example, R may bind to another protein. That's the ribonucleus. So this ribonucleus will cut RNA, and the RNA will disappear. So similarly for protein. So in that case, we can also consider other reactions for autoregulation. So in that case, the process would be more complex. So here we don't have a discussion for that. So next we consider mathematical modeling. So that means, for example, if we have a system, we may have a number of reactions looks like that. So the question is how to get mathematical equations based on all these reactions. That's the next step for mathematical modeling. So if we consider mathematical model, we have different types of models. For example, we may consider a graphic model. That would be a very simple one. We just consider what's the relationship between the molecules. We may consider Boolean equation. Boolean equation, the value of variables will have only two values. There's 0 or 1. Boolean is a simple one, but it can be used to represent a complex system. So we may also consider some linear or nonlinear equation model. We can also consider differential equation models. So in that case, we may have different types of mathematical formulas to describe the chemical reaction system. So if we consider differential equation, so we may have a different formula for the differential equation. For example, linear equation, neural network, and also shear arc models. The third one is a quite popular one for genetic regulation. S system is another type of system. Is normally used for the metabolic systems. So here we consider the general idea for mathematical modeling. So as we mentioned in the previous slides, so for example, we consider these slides we mentioned when we have a detailed understanding of biological systems, we may have a number of reactions. So generally, you can also get, there's use some molecules to get some other molecules. So in that case, these molecules will be consumed. It will disappear. And then this is the product of the molecule. This molecule will in the system. So in that case, if we consider all the reactions, we consider we have a number of reactions. That's R1, R2 to Rm. We have m reactions in the system. And then we have n variables. That's x1, x2 to xn. So that's all the variables inside the system. So in that case, we may consider this xi is a molecule number. So if it is a molecule number, so this should be integers. Or this variable may be concentration of the variable. So in that case, it should be a real value. So this is the structure of the network. And then for each reaction, so as we mentioned, we normally consume some variables. After that, this variable will disappear. And then we get the product for xm and xn. So these variables will inside the system after the reaction. So here K is the reaction rate. So it represents the fast or slow of this reaction. So if we have a detailed reaction looks like that, we can define a propensity function. So this propensity function, that is the probability of this reaction will fire. So generally, the propensity function can be defined as this is the reaction rate, K. So this is a reaction rate times variables involved in the equation. So this you can consider, okay, that's the variables on the left-hand side. So because this variable will disappear, you can say if the reaction rate is large or any variables have a very large number for xi and xj. So that means this reaction would be more possible to happen. So in that case, this propensity function would be larger. So generally, for propensity function, we will consider, that is the reaction rates times variables on the left-hand side. So that's the basic principle. So this is the probability. We also need a stoichiometric vector to represent the change of the variables. So in this case, because xi and xj will disappear, to get a new molecule xn and xm. So in that case, I and j should be less than 1 because it will disappear. So it should be less than 1. On the other hand, xm and xn should be increased by 1. So I think this is the basic idea, to represent a chemical reaction. Okay, so here we consider this example. We have a system with 4 reactions. So first we can define 4 propensity functions. So as we mentioned, you should use the reaction rates times variables on the left-hand side. So you can say this is you can get the results for A1, A3, and A4. For A2, it is different because we need to use 2 same variable x1 to get x2. So in that case, you may have classic probability problems. For example, in a bag, we have X red balls. So we first grab a ball out. So in that case, we have X1 possibility. After that, the ball inside the bag would be X minus 1. It's not X again. So in that case, if we get the second ball, the possibility is only X minus 1 here. And then if for these 2 balls, if we don't consider the order, any one may be the first one. So in that case, that's the combination problems, it should be divided by 2. So this is the idea why we use this formula to define A2. If, for example, this reaction is X1 plus X3 to get X2. So in that case, X1 is different from the second variable, so we don't need to consider that. So in that case, there would be only C2 times X1 times X3. So this is a special case if we have 2 same variables on the left-hand side. So this is the propensity function. And then if we consider the stoichiometric vector, so that is the change of the variables. For example, for the first equation, x1 will disappear by 1. x2, x3 unchanged. So in that case, only x1 minus 1. x2, x3, no change here. So similarly, we use 2 variable x1 to get 1 x2. So we define this new vector. So in that case, we can define 4 vectors for these 4 reactions. So if you can understand these slides, so it would be relatively simple to get the following equations. So as we mentioned before, the next question is how to from all these chemical reactions to get differential equation. So in that case, we have already defined the propensity function here, defined the stoichiometric vectors here. So in that case, we can simply put all this together. For example, here we have 3 chemical reactions. that's the predator questions in ecology. So in that case, x1 to get x2, get 2x2, that means that we have a new species for for y1, and then y2 will eat y1, and then have a new y2. So that means y2 will eat y1, and then they will have a new baby, and then they have a new species of y2, and the y2 may die when it is old. So this is the 3 reactions. So similarly, we can define these 3 propensity functions. So for example, that is, if we consider here is K1, K2, and K3, you will find the first equation would be K1 times the left-hand side of Y1. The second would be K2 times Y1 times Y2. So this is the 3 propensity function. And also, you can define 3 vectors for these 3 reactions. So here, that's the Y1 will get a new one. The second reaction will use Y1 to get Y2. The third one is Y2 will disappear by 1. So in that case, this is the vectors here. If you consider the multiplication of these 2 vectors, so you will get this equation. So here you can see, from the chemical reactions, you can get the ODE system. So the intermediate step is you need to get 3 propensity functions and also get 3 these vectors. This is the one. And the second one, for example, if we use 2 variables for P2 or 2 variables P to get the new variable P2 here. So that is the case, there's a P plus P to get a P2. So in that case, so similarly, because if we consider the concentration, so in that case, when we consider concentration, we don't need to define the, like the previous slides here, we have to consider x minus times x minus 1. In this case, we consider that this is the molecule number. If we consider the concentration, the propensity function is simply this way. You may have a question, so why the second one is fine. If we consider the vector, that's fine. We use 2 get 1, so that's the first variable minus 2, the second variable plus 1. So this is okay. The second propensity function is okay. But why for the first propensity function? So here, as I just mentioned, here we consider x1 times x1 minus 1. If we consider concentration, we count minus 1. So we simply consider x1 squared. But here we have a half. Where is the half now? So actually this, in some sense, my understanding is you can combine this half to the reaction rates to get a new reaction rates. So in that case, you don't care about whether you have a half here. Anyway, you have a reaction rate. You need to estimate these reaction rates. So it doesn't matter. So in that case, we can consider K1 here. So we can get these 2 equations. So here I just give you some. This is an example when you can read a different textbook. people may use different notation, use different understanding to represent an equation. So this is the case for the modeling. So certainly, if for the systems with 4 reactions we just discussed, so we can consider here. So this would be, 4 propensity functions, and then we have 4 vectors, and then when you multiply these 2 equations, these 2 matrices, we will get a system of ODE system to describe the dynamics. So here, if you use, x1 minus 1, the underlying assumption is variables in the system is a molecule number. If you consider concentration, as the previous slides, if you consider concentration, you count -1. So that is if you consider molecules, you can -1. If you consider concentration, here should be x1 squared. So if we have ODE systems looks like that, so we can use MATLAB to get the simulation. So that is the case, for the system we just discussed. So the idea is, for these chemical reaction systems, we first formulate chemical reactions, and then we consider propensity functions and stoichiometric vectors, and then from propensity function we get the differential equation, and then we get the simulation. Actually, for stochastic simulation, we don't consider the differential equation. So that's the next topic for stochastic simulation. So before we consider stochastic simulation, we need to discuss 2 types of noise. There's the external noise and internal noise. So internal noise, that's the noise inside the system. Generally, this noise is generated from molecules with very small number. So if, for example, if we consider gene expression, the number of DNA will only 1 or 2. So that means you can immediately realize if the transcription process start, then the molecule number will change substantially. If the transcription don't start, it keep quiet, so the molecule number will be quite small. So in that case, protein with small copy numbers may generate variations in the system. That's the internal noise. External noise, you can consider that's the noise outside the system, but this noise may influence the system we are interested. For example, if we consider chat box, okay. Okay, so if we, okay, thanks. So if we consider transcriptional factor, for example, for transcriptional factor, the number may change, but the transcription factor is outside the, that's generated from other system. You can consider is a molecule outside the system. So it has influence to the gene expression. So there's the 2 types of noise. To describe the 2 types of noise, we may use different equations, different mathematical formulas to describe the noise. For example, we may use chemical reaction system. Chemical reaction system is for internal noise. Stochastic differential equation normally is for external noise. We may also consider, model with other types of stochastic process. For example, for Poisson process. So in that case, we can use that for either internal noise or external noise. Doesn't matter. So anyway, certainly for stochastic differential equation, we can also use it to represent internal noise, but we should have certain restriction for that. Otherwise, the accuracy wouldn't be good. So again, here we consider the chemical reactions again. We have, defined the propensity function and the stoichiometrics before. So in that case, we have a number of 4 reactions. We can define 4 propensity functions. So here we have, 4 vectors here. So if we consider stochastic simulation, so the basic idea is we assume the time step would be quite small. And then in this time step, there is only one reaction file. So that's the basic idea for stochastic simulation. So that means if we consider time point from t equals 0, that's the starting point. We may consider the first time step is t1. And then in this time step, there is only one reaction file. And then from T1 to T2, so in this time step, from T1 to T1 plus T2, so the step size is T2. So in this time step, again, there is only one reaction file. So in that case, we can, we needed to find 2 values for the stochastic simulation. The first one is, what is the value for T1 and T2? So, or how to determine the step size. This is the first question. Okay. The second question is, if we determined in T1 step, we have only one reaction file. However, in the system, we have 4 reactions. So which reaction from these 4 reactions will fire? So that's the second question. Okay, so if we have determined these 2 questions, for example, in the first step, the time step is T1, and then we assume the second reaction will fire. So for all these 4, the second reaction fire. So in that case, we use this is the molecule numbers, that's the initial molecule number. So that's all are 100. Because this is the second reaction fire. So in that case, we needed to use the second vector. The second vector plus the original X vector, and then get the result. So in that case, X1 will be decreased by 2, X2 will be increased by 1, X3 unchanged. So similarly, in the next step, if we determine the time step size is T2, so we get the time to T1 plus T2. And then we determine that, that's the 4th reaction file. So in that case, because there's a force reaction, we needed to use the force vector to plus the variable vector here. So you will see x1 unchanged, x2 minus 1, x3 plus 1. So that means, so in the following one, that would be relatively simple. You just needed to use the current variable number to plus the vectors of that reaction. This computation is relatively simple. The question is how to determine the step size T1, how to determine the reactions file in this time step. Okay, so in that case, we needed to consider the exponential random variable. So when we learn probability, we know exponential random variable can represent the so-called exponential waiting time. So for example, if you go to a bus stop and waiting for a bus, the waiting time follow an exponential distribution. So in that case, if we consider the average waiting time is AX, so that means the random variable will follow exponential random variable. The parameter is 1 over AX. So that's the so-called exponential waiting time. So that means if we consider, because the average the waiting time is 1 minus the 1 minus A. So that means if the propensity function is large, so in that case, the waiting time would be smaller. So in that case, we can use this exponential random variable, and then you can consider this is the probability. And then we can use a sample from the uniform distribution between 0 and 1. We can let this sample equals the waiting time, and then we can get the values for that. So that is the equation, original equation, R equals the exponential values. And then from this equation, we can find the dt here. So this dt actually is the waiting time for the next reaction. So this dt actually is T1, T2 in the previous slides. So this is for step size of the next reaction. In the stochastic simulation algorithm, that's Professor Gillespie developed in 1977. In the original paper, he proposed two methods for stochastic simulation. The first method is we discussed in the previous slides. So that means we determine a time step and then determine which reaction will fire, and then update the system. That's the so-called 3 steps. It also, we also have another simulation methods. This is a different one. So that means it will determine the waiting time of all the reactions. If we have M reactions, we need to determine M reaction time. And then we will see which reaction is the fastest one. For the fastest one, that must be happen. So in that case, we consider select reaction based on all the reaction time. That's a different one. But anyway, the simulation result is nearly the same, but the steps is different. The point is for the second methods, normally we need more simulation time. For the first one is relatively simpler. So that's the method widely used in application. So for this one, so as we mentioned, so in that case, in the Gillespie's method, we first sum all the propensity functions. So if we consider the reaction for the system, we needed to first sum all the propensity function, and then from this propensity function, we determine the step size. So if we consider okay, so if we consider this one, actually this is a reaction for a particular, this is the time step for a particular reaction. If we consider there is so-called A0, that's the sum of all the propensity functions. That is the time step for the whole system. So that means in this system, we have only one reaction file. So that's the assumption for that. And then we need to consider, which reaction will fire. So in that case, as we just mentioned, if we sum all the AJ together, that's the value for A0. So in that case, this A0 may be small or maybe large. So we first need to change all the values to unit 1. So that means we divide AJ by A0 to get a new variable for UJ. So it is clear if we sum UJ together, the value should be 1. Okay, so with that, so that means from 0 to 1, if we consider this interval, we can divide it as a number of subintervals. And then the length of the first subinterval is U1. And then for the second subinterval, the length actually is U2. And then for the third one, the length is U3. And then you can see, okay, so actually by all these subintervals, the length of each subinterval is U1, U2, U3, and then to UN. But all the subinterval together, it is from 0 to 1. So in that case, we can generate a sample from the uniform distribution. This sample, the value is from 0 to 1. And then we can see, okay, this sample in which subinterval? If this sample, for example, in the third subinterval, that means the third reaction will fire. So that's the idea to determine which reaction is fired. Actually, this idea is widely used in statistics for importance sampling. So that's the similar idea for that. So in that case, we can determine which reaction will fire in the given time step. Then, that means if, as we mentioned before, we have 3 steps. The first step is to determine the step size. The step size is based on A0. A0 is the sum of all propensity functions. We determine step size. The second step is determine which reaction will fire in the time step. And then if we determine it is the L reaction fire in this step, we will use the vector of the L reaction to update the system. So this is the idea, for the stochastic simulation algorithm. So in that case, if you understand the propensity function, Aj, understand the stoichiometric vector Vj, so that's the first starting point. You have an understanding for biochemical reactions. And then mathematically, you need to determine the step size, determine which reaction will fire, and finally, you update the system. So this, generally, this reaction needs a huge computational time. If the system is large, we can't get, it would be difficult to simulate that. So this is the MATLAB code for simulation of the system before. So you will see, okay, so this is the 4 reaction rates. There's C1 to C4. So V is the stoichiometric vector. So actually this vector, that's the vector, okay, that's the 4 vectors here. This is the vector. So this is the 4 vector. So we have a initial value. This is the simulation time for capital T. So that's the next for 400. And then in each step, we will generate 2 samples. That's the uniform distribution random variable samples. So we needed to calculate 4 propensity functions. That's the formula. And then we get sum all the variables together. Okay. And then that tau is the step size. So that's the formula we discussed in the previous slides. Is 1 over A0 times log 1 over R1. So that's this formula to determine the step size. And then we needed to determine which reaction fire. So basically we will plus, like the slides here, we will plus Uj values 1 by 1. And then determine the second sample satisfies these equations or not. So that is, in that case, we sum the sample 1 by 1, and then we determine whether the sum is greater than the standard value. So that's the standard value is sample times A0. Here we don't consider Uj, we directly simplify the computation. After we determine the index as the next reaction, so we just needed to update the variable, update the time, and then save the simulation result. So this is the MATLAB code for the simulation. So if you have other systems, you can use the same structure. You only need to change the vectors here, change the propensity function here. You can get the similar simulation, code. So this is the simulation based on stochastic simulation. So it's similar to the ODE result, but you will see fluctuations in this case. So this is the stochastic simulation. In the second one, as we mentioned, in that case, we needed to determine, in the second SSA, we needed to determine all the reaction time for each reaction. So that means we need to use AJ to determine step size TJ, DTJ. And then we need to, for each reaction, we need to generate a sample. But in that case, we need to check whether AJ is 0 or not. If AJ is 0, so that means this DT would be, is impossible. So that would, you can assume DT as, 1,000 or any larger number. That's impossible. Okay. And then if we determine the reaction steps for each reaction, we can find the minimum step size. So we find that this minimum step size, the index is I, so we can, update the system by the ith vector vi. So that means, so this is the second step, SSA. If you are interested, you can use the similar code in this one to write a code for the second SSA. So as I mentioned, generally, this method is more popular because it's more efficient. For the second one, you can see we not only need all these samples, we need to calculate all the step size. This computation is large if there are more reactions in the system. So this is the second SSA. So SSA, this method is was proposed in initially it's 1976. It is mature in 1977. So normally we consider 1977 is the starting point of SSA. However, after more than 2 years, it has only 30 citations. So actually no one used that method because the computational time is very large. Actually, initially, this method was criticized until in 1998, Adam Arkin in Berkeley proposed the first stochastic model to simulate a lambda fudge system. This method, this paper can be considered as the starting point of stochastic modeling. So in that case, SSA has used widely. Now the citation, my impression is more than 10,000. It's quite a large citation now. So that's for SSA. But after Adam Arkin's first research work, people realized SSA needed much time. The point is we needed to determine in each step, we needed to calculate the propensity function, determine the step size, then determine which reaction file. It's quite time consuming. So Gillespie then formulated the first method. So that's the so-called Tau-Lip method. The idea is in Tau-Lip method, we use a relatively large time step. And then in this time step, there may be a number of reactions inside the step. And then we needed to use all these reactions to update the system rather than one single reaction in the SSA. So that's the difference between SSA and the Tau-Lip method. So the idea is, for example, we have 3, this is the example, we consider a relatively large time step. There are a number of reactions. In the system, we have 3 reactions. If we count the time, so we will say R1 has 5 times, 5 reactions. R2 has 2 reactions. R3 with 3 reactions. So that's the number for all the reactions inside that. So in that case, Gillespie assumed this reaction number, that's the 522, is the sample of the Poisson random variable. For the Poisson random variable, the mean is the propensity function times the time step. So for the 3 reactions, we use A1, A2, A3. So that's the propensity function. Then times the time step tau here. And then with these 3 Poisson random variables, we can generate a sample. Then sample would be the reaction time in this large time interval. So that's the case for Tau-Lip method. So for Tau-Lip method, actually you will see, if we consider the one, if you determine the step size, the key point is the step size. If you determine the step size, you can formulate these 3 Poisson random variables. So you have no difficulty. You just use A1 times tau1 times tau. So consider this one. And then in the following step, you can generate a sample. And then based on sample, you can update the system. So in the Tau-Lip method, the key point is to determine the step size. So in that case, that's the method here. So we first need to determine the step size, then generate a sample from the Poisson random variable. Then based on the sample number, this is the sample number. You can use the sample number times Vj to update the system. So this is the computational step for Tau-Lip method. The key point is how to determine the step size. For step size, we have a so-called Lip condition. The Lip condition is, in that case, we hope the change of system wouldn't be very large. So actually, I think this is the similar to any problems in computational mathematics. Computational mathematics is always a balance between the efficiency and the accuracy. If the step size is large, it is efficient, or we can get the results very quickly. On the other hand, the accuracy is not good. I think that's the same as the Tau-Lip method. So we can use epsilon here to determine the step size. If epsilon is small, step size is small, it would be more efficient. If epsilon is large, we can get the simulation quickly, but the accuracy is not good. There's the same problem for that. So the point is, here we just have a discussion for the so-called Lip condition. So the Lip condition is, as we discussed in the previous slides, we hope the change of the propensity function is not large, restricted by epsilon times A0. So in that case, if we consider, each variables here, if we consider the Poisson Tau-Lip method. So as we mentioned, the xi should be, use the Vj factor, the i's for the ith element times the sample. So I think that's the this is the expression for this one. This is for the vector form. X is a vector include x1, x2 to xn. If we write it, consider as the element form, that would be the element form for a single variable for xi. And then if we consider this is the increment, that's the change of the propensity function. So we can use the average plus the standard deviation or minus the standard deviation. We can use this one to represent that. Because actually, because X is a random variable. So that's why we use the mean and the STD to represent that. And then, we consider the mean of the increment for this one, because in this case, P is a Poisson random variable. So there's a stochastic. So if we consider the mean of this one, we don't consider it as a random variable. We only consider the mean. For Poisson random variable, the mean is the parameter. So we can get these formulas for the increment. So this is a deterministic representation of the Poisson tau-Liebel method. And then we consider the Taylor expansion. So that is the AJ, the initial point is xt, the increment is delta x. So we consider the Taylor expansion, only consider the linear term. Then we have the first partial derivative, and then if we write the first partial derivative here, so we can get these new variables. So that means if you consider this equation, that means the increment, that is AJ, the left-hand increment in the previous slides, the increment is AJ xt plus tau minus AJ xt. So this is the increment. So from here, so if you substitute, remove this term, you will see actually this is the increment term. So this increment term should be less than epsilon times A0. So in this case, this is the idea to determine the Liebel condition. From this equation, you can find the solution for tau. So that's the tau for each reaction. And then if we consider all the tau for each reactions, we find the minimum step size. That's the step size, for the tau-Liebel method. So this is the idea for tau-Liebel method. Actually, for this method, this is the initial method proposed in 2001. But later, Gillespie realized that method is not good enough because the step size varies much. So in later, he proposed a better method. So in that case, they consider this is based on mean, this is based on standard variation. So that's the get a more restricted step size. The step size is smaller, but the accuracy is better. So that's the case for improved step size process. Actually, Gillespie and Yang Chao at Virginia, they also consider more restricted form. You can see here, actually, the computation actually is more and more difficult. Actually, it's not easy to determine the step size. So that's the case. So that's why we, Gillespie determine the step size more carefully. The problem is a so-called negative simulation number. So actually, the negative simulation number was found by Kevin and me in 2003. So when we first calculate the system, we find if we have a relatively large step size, it is easy to get the negative number. So here we consider an example. So in this case, we use X1 and X2 to get a new molecule for X3. So if we assume reaction rate is 1, certainly is quite large. And then X2 is small, but X1 is small, but X2 actually is very large. So in that case, if we consider the propensity function, that would be K times X1 times X2. So in that case, we will have a Poisson random variable with mean 20,000 times tau. So no matter what the step size here, it may be quite easy to get a sample. The value would be 3. The sample would be 3, or even larger than 3. So in that case, because X1 only have 2 molecules, if 3 reaction fails, then it would be X2 minus 3, we get a negative 1. But in reality, actually all the molecule numbers should be positive. At least, at most, it may be 0. It can't be negative number. So in that case, that's the problem. So you may say, okay, in that case, we may use a very small step size, tau here. Okay, so if you use a very small step size, that will only reduce the probability to generate negative number. It's still possible to get a negative number. So that's the problem for that. So that means if you, for these systems, if you have 1,000, 10,000, 100,000 simulations, you may finally get a negative number. So that's the point. So we can't avoid such a situation. So in that case, we propose the binomial random variable. So binomial random variable, so in that case, there's n Bernoulli test. In each test, there will be with a probability of 0, probability of P to get the result 0 or 1. But in that case, the maximum number, if you consider binomial random variable, the maximum sample value is n. It can't be n plus 1. So in that case, this would be a restricted, sample value. So that's why, in order to solve the problems of negative number, we propose to use binomial random variable for that. So for binomial random variable, the key point is what is the n? Because n times p should be the propensity function. Because in that case, the probability, so the propensity should be the same as the Poisson random variable. So in that case, if we consider these, reactions, we need to determine which one is n, because x1 is small. We hope to restrict that, so we use x1 as n. So if consider original propensity divided by x1. This the remaining one is the probability of P. So in that case, we get a binomial random variable. So certainly for binomial random variable, the probability P should be less than 1. So in that case, we have a restriction for that. So there's a restriction for the step size top. So in that case, if for each reaction, we use a value with small number as n, and then use aj divided by xk as the probability, so we can generate, binomial sample and then update the system. It is clear for this, if the system is relatively simple, so we can guarantee all the simulation results is non-negative. So here we just need to carefully choose xk for each reaction. But certainly, problems is more complicated than this simple one. So, for example, we consider the next reaction. So in that case, S2 involved in 2 reactions. The first one is involve S1 and S2 to get S3. But S2 itself can produce X4. So if we use the binomial random variable in the previous slides, we can guarantee for each reaction, the reaction number is less than 2. That it doesn't matter. However, if we consider the total reaction number of S2, it may, the reaction number may greater than 2, we may get a negative number. So in that case, in this case, we needed to determine the total reaction number of these 2 reactions. Because this is for 2 reactions, we needed to consider A1 plus A2 as the propensity function. And then use X2 as n to restrict the reaction number. So once the total reaction number is determined, so we can determine the reaction number of the first reaction based on the total number. And then for the number of second reaction would be, the remaining one. So in this way, we can determine the reaction numbers for this case, and then we can guarantee for these 2 reactions, after this method, the simulation time is positive or non-negative. However, the question is, here is we consider S2. If S1 is involved in the other reactions, so you will say, okay, this would be quite complicated. So in that case, it is, in my opinion, it is still an open problem to guarantee positive simulation values. So that's the one. That's why in 2006, Gillespie proposed, other simulation, the method to determine the step size. So that is, he hoped to determine a proper step size and then guarantee the positive simulation. So that's the idea, we consider the Tau-Leap method. So in that case, we proposed binomial Tau-Lipper method. That's the whole method we considered. So this one, the first step, we needed to divide the system into a number of subsystems. So we needed to find the values of the reactions that may be independent reactions. That means the reactions don't share any variables with other reactions. That's independent reactions. Or we need to consider group reactions if a molecule involved in all these reactions. So if we consider for the independent reaction, we can simply use the binomial random variable to generate a sample. That would be quite simple. However, for group reactions, we first needed to sample the total reaction number of the group, and then based on the total reaction number to determine the reaction number of each reaction. So that's the binomial Tau-Lipper method. So this is the one Tau-Lipper method. A lot of binomial Tau-Lipper method. So due to the time, we don't hope to discuss here because this method actually introduced bias. We ignore that. Another one is so-called R-Lipper method. R-Lipper method, that's a different idea there's a different idea from the Poisson Tau-Lipper method we proposed before. Actually, the idea of R-Lipper method is also proposed by Gillespie. If we consider the Poisson Tau-Lipper method, if we consider this picture, okay. So the Poisson Tau-Lipper method is first determine the step size. So for the step size, it is a deterministic one. We use some deterministic method to determine the step size. And then in this time period, what is the reaction number? We don't care. This reaction number is determined by random samples. So in some sense, we can say, okay, the step size is a deterministic one, but the reaction number is a stochastic one. So for R-Lipper method, there are a different idea. So he first determined the step size. The step size is a deterministic one. For example, we only consider in the next step, we consider 10 reactions. Okay, we don't consider too many. We only consider 10 reactions, or only consider 5 reactions or 20 reactions. The reaction number is fixed. We can, fix a value for that. So the question is, what is the time period to realize these 10 reactions? Or what's the value of step size tau? It is determined stochastically. So that's the different point. So actually, well, for example, in the Tau-Lipper method, we can determine, okay, step size is 0.1. We fix the step size. The value is 0.1. And then in this time interval, how many reaction files? We don't know. We use sample to determine that. So that's a different idea for Tau-Lipper method. So in that case, he first, fixed L reactions. So it is first determined. For example, as I just mentioned, we hope in the next time step, there are 10 reactions. So in that case, with 10, with the L reaction, we can use gamma distribution to determine the step size. So the parameter would be L reaction, another is 1 over A0. Now, that's the A0 is the sum of all the propensity functions. And then we determine the step size. Okay, so the next question is, for L reactions, we need to determine, for the first reaction, there are how many reactions. For the second reaction, there are how many reactions. We need to determine all these values. So in this case, the idea is similar to the binomial random variable. So we first, based on L reaction, okay, we based on L reaction to determine the number of first reaction. So that's K1. After K1, the reaction number for the remaining, that's from R2 to Rm, should be L minus K1. So in that case, we use this binomial random variable to determine the reaction number of the second reaction. And then you can repeat this process, determine all the reactions there. So this is the R-leap method is similar one, but certainly for R-leap method, we may also get a negative number. So in that case, he also proposed the techniques to avoid a negative number. But anyway, this is two types of method to accelerate computational time. Here is a chemical master equation. So we have in this case, if we consider stochastic simulation, it is the direct simulation of the system. What's the change of molecule numbers? So in that case, if we consider the probability of the simulation result, for example, when you get the result, you get the hist, that's the plot of the results. Actually, that's the distribution of the random variables. So in that case, if we consider the probability of system at X state, when the time is, this at a system is at a state X at a time T. So we use this value to represent the probability. So in that case, if we consider all the probability change from T to T plus dt, so we consider the probability, that's the change of the probability. So in that case, actually we have, this is the case, we have the following three cases. So in that case, if we consider that's the, actually that's for xt at the time t. Actually, this should be xt plus dt. If there is no reaction fires, the system is the same. So we can keep the probability. The first case is if the system state is xt plus vj. And then in this time period, from t to t plus dt, if the J's reaction fails, So if you consider the system state plus Vj, so actually this will cancel. So we can keep the state as xt. So this is the case, if the system is x minus Vj, if we have a reaction, so that will from other state to the current state xt, and then the probability will increase. However, if the current state is xt, but the Rj reaction fails, because Rj reaction fails, the system will move to xt plus Vj, or in that case, it moves from the state xt. So in this case, the probability will be decreased. So in some sense, this reaction represents 3 cases. There's the no reaction. There's a reaction from other state to xt. That is the reaction from xt to other states. So the probability will be decreased. So for this reaction, that means if we consider, move the pt, this term to the left-hand side, okay, and then divide dt. Let dt approach 0. That's the standard definition for derivative. So in that case, if we consider this computation, we will get the following equations. So in this case, you can see the left-hand side is a partial derivative. The right-hand side would be, that's the propensity function times the probability. So this is the so-called chemical mass equation. The remaining one is just some notations for the chemical mass equation, because we don't consider linear noise approximation. We don't consider that one. We directly move to the next slides. So in that case, we multiply x, to both sides, and then get the sum for all the cases of x. So that's the case for operation for the chemical mass equation. So for the left-hand side, so you can see we can move x inside the derivative. So we get this one. For the right-hand side, actually, when we have 2 sum notations, I think generally the first step is to change the order of the sum notations. So we put j to the outside, then consider x inside. So that's the case. In that case, if we consider x times aj, x minus vj, because all this is x minus vj, we should match x minus vj here. So if we minus vj, we should plus 1 vj to get the equation the same. So this is the first part. Actually, the first part equals to these 2 parts. This term is unchanged. We just put x inside that. In that case, you can say, actually, this is the notation for the mean. So if you consider the mean of the random variable is x times the density function. If you consider the mean of a function, it is the function times the density function. Okay, so in that case, you can consider this is the density, pxt is the density function. So in that case, actually, this notation represents the mean of X times AJ. So if you remove all these X minus Vj as a new variable Y, you will say, actually, that's the same mean as the second one. So in that case, these two mean can be canceled. On the left-hand side, that's the mean of X. So in that case, you can consider that's the derivative to the mean of X. That's the left-hand side. Okay, for the right-hand side, the mean of these two terms canceled. So we only have this term, but Vj is constant. We can move Vj out, doesn't matter. So inside that, we only have Aj times the density function, Pxt. So that's the mean of Aj. So finally, we can get this rate equation. So this is the step from the so-called this chemical mass equation to the rate equation. So for this one, we can get the following formula. I like this formula because this will give us, the information for all the connections of different simulation methods. Okay, so here we don't write the SSA because SSA is you can consider the left-hand side equals the right-hand side plus only one reaction plus Vj, only one. Okay, that's the SSA. In Taulippo method, we may have a number of reactions, and then we need to consider more update steps. So here, the point is how to simplify the Poisson random variable. So as we know, for a Poisson random variable, if the mean is large, if the lambda is large, so in that case, the Poisson random variable can be approximated by Gaussian random variable. The mean and the variance is the same. They all are equal to lambda. So for this Gaussian random variable, we can separate lambda out and then consider the standard Gaussian random variable, or we put the variance out. So that's the case for the approximation of the Poisson random variable if the mean is large. So if we rewrite the Poisson Taulippo method, so in that case, we can, this is the mean, we can get the lambda here. This is the variance times the standard Gaussian random variable. So these formulas you can consider that's the approximation of the following stochastic differential equation. So for example, if you put square root of tau times the standard random variable n0, 1, in that case, this is the increment of the Wiener process. And then this is dt. You move this term to the left-hand side, so this is dt. So this is the deterministic term, you change tau to dt. So this is you can consider that's the Euler method to solve the so-called chemical Langevin equation. There's a special stochastic differential equation. For this stochastic differential equation, if we assume the Aj is large, so you can consider this is the second condition. If Aj is large, so Aj, then if you consider the square root of Aj, the value would be much smaller. For example, if Aj is 10 to 4, so this is a very, very large value, 10 to 4. If you consider the square root is only 100. So in that case, this only this small value can be considered small fluctuations can be neglected. We don't need to consider that. So in that case, we only have the deterministic part, then we get the ODE equation. Actually, the ODE equation is the rate equation in the previous slides. They are all consistent. So actually, in these slides, we have 4 formulas. The first SS1 is the SSA. We didn't write that. That's the second equation. That's the Poisson-Taulipo method. And then that's the chemical Langevin equation. That's a stochastic differential equation. And finally, is a rate equation. That's the ODE equation. So if we consider all these 4 different forms, actually, we can find the application of this one. For example, if we consider the chemical Langevin equation, the condition is lambda should be large. If lambda is not large, this approximation is not valid. That's the first condition. The second condition, if we use ODE, that means Aj should be large. If Aj is small, so this value, the fluctuation can't be avoided. We must keep the fluctuation. So this is the equation for that. Finally, so we consider some models for genetic regulation. So actually, if we consider these 4 reactions based on the central dogma, that's from DNA to mRNA, and then from mRNA to get the protein, and also there's the degradation of mRNA and the protein. So if we consider SSA or Taulipo method, as we discussed in the previous slides, that's the direct simulation. That's the one model for that. And we can also consider, there's the ODE model. If we consider direct, from the equation, if we directly consider the rate equation, only consider the rate equation, that's the Vj times Aj. Consider with rate equation, that's the ODE model system. This is the second one. And the third one is we can use a Poisson random variable. That's the method we proposed before. In that case, we can, you can use method to use Euler method to find the solution. But if we don't consider the Poisson random variables here, that's the Euler method. But for Euler method, for each term, we use the Poisson random variable to represent that. So in that case, we have a stochastic model with Poisson random variable. And this one is from this equation, we can simplify the system. To simplify the system, we consider this the so-called Faster equation or slow equation. If we, for example, we assume the first reaction is the fast reaction, that means it reaches the equilibrium state, the derivative of m should be 0. If the derivative is 0, so from this equation, we can directly find the value of m. Then substitute this variable to the second one, we can get the equation for that. We can get a quite a simple equation for the whole system. Certainly the question is, what is the fast reaction? What is the slow reaction? We need to make the assumption based on biological problem. And then we can also consider stochastic differential equation. So that means in the original ODE model, we introduce noise term directly. We can consider additive noise or multiplicative noise. We can use different terms here, or we may consider chemical non-general equation. Chemical non-general equation is a special one. So we consider the square root of the terms. Okay, so I think the following we have some examples, but I don't think we have time. Rather than I have all the slides there, we may have a discussion later. If you can send me an email, and I think the discussion, my presentation stop here. Thank you for your attendance. So next we have some discussions.
Okay, thank you, Dr. Jian. But I'm wondering what the result of this. Do you have the slides of the numerical simulation?
Yes.
For this example,
We continue to finish that part.
Just show quickly how it looks like, maybe.
Okay. So okay. So this is a system for the DNA damage system. It was published in Nature in 2000. This is the first synthetic network. So he used the ODE model to describe the system. So in that case, you can consider that's the increase or decrease of each variable, but they use a term here to represent the intervention of the DNA damage signal, MMC. So for this one, if we consider the simulation of the system, we can choose different value of S1 to represent the strength of DNA damage signal. So initially, as you know, in the simulation, U is large value. So if we introduce S1, U will be decreased, and then V will be increased. And then, in this time period, the system will change. And then determine, based on different values of S1. So in that case, we can, the system may not change to the next state. So that's called a genetic switching. So in that case, if the signal disappears, the system will return to the original state. So that means for example, in 50 to 900. So that's in 50 in 900, S1 is non-zero. After 900, S1 is 0. So that's the mathematical realization. So if S1 is 1.7, so in that case, the change is not large, system will return to the original state. If S is very large, is larger, so the system will change to the different state. So that means U will change to the low state, V will change to the high state. So it depends on the value of S1. In deterministic simulation, we have only one result. So depending on the S, it may be return to the original state or change to the other state. However, if we consider the biological experiment, you will see actually when we use different DNA damage, signal MMC here, actually it means part of the molecule, part of the cells will change to the other states. Unlike here, if no signals, okay, so actually all the cell in the first state, if the signal is getting larger and larger, then would be more and more cells move to the second state. So it's not 0 or 1. Actually, it represents a certain probability. So to realize that, so we propose a stochastic model. So the power of the stochastic modeling is if we have the same model, same parameters, we may realize different simulation result. That's the same parameter, but in these situations, the cell may switch to the other state, or the cell may remain in the original state. So if the value of S1 is getting larger and larger, that means these two states will be getting closer and closer, and then it's more possible to switch. So in that case, if we have different value of S1, that means different cells will switch to the other states. If the S1 is very large, that means all the cells will switch to other states. So I think that's you can say this case is similar to the experiment result. That's the first model system. For the second one, that's the ecological.
Yeah. Yes. I think we can stop there for a moment, Dr. Tian. I will ask the audience if they have any questions or need some more explanations.
Okay.
Audience? Yeah, because we only have like 30 minutes remaining. Do you have any questions? I think the material is interesting. It's new to us, Dr. Tian, because we are not into the biological system modeling. We have courses stochastic calculus. We have modeling in stochastic differential equation, but not in the application in the biological system. So we have, for example, for a spread of disease like that. Yeah, it's still simple. Yeah. So here, I'm also learning from you here because it's also new for me. I think this gene expression network is quite difficult, yeah, to model. So what I catch from your lecture is how to determine the step size. That is the key point, right?
Yeah.
That is the stochastic is in the determining the step size. So you have that tau leaf using Poisson method, binomial method, and R leaf with the gamma distributions, a combination. I mean, you have to have a good basics on statistic mathematics also.
Yeah.
I was thinking, yeah. What about the parameters? It's also difficult to determine or estimate the parameters. So in this case, how do you determine the parameters?
Okay, yeah, that's yeah, you are right. So, for example, for this slides, we need to determine the value of epsilon, because the epsilon, the value of epsilon determine the step size of tau.
Yeah.
So in our simulation, so here we don't have any, how to say that? We don't have any formula for that. For example, when we consider simulation of SDE, so the step size may be 0.1, 0.01, or 0.05. So we have some impression. So if we hope to get a good result, the step size should be 0.01 or even smaller. So in that case, we only from the simulation, we say if epsilon is 0.5, so the error is a little bit large. If epsilon is, I didn't, maybe 0.03. Let me check for my original public. So I think it should be 0.03 or something like that. Yeah. Yeah, 0.03. 0.03 is fine. If epsilon is 0.01, the step size may be quite small. So the efficiency. So in this case, I think that's a similar one. If you use a small step size, that means the computational time is large. If you use a larger step size, the computation time is okay, but the accuracy is not. Yeah. So normally, I think similar to the other problems, we depending on the system, depending on the computer on my desk. If the computer is larger, I can use a smaller step size. That's the same. Yeah, actually there is no formula for that.
Yes. Yes. So yes, of course, there's a like trade-off balancing between efficiency and accuracy.
Yes.
But my question was the parameters for the reactions. I mean, like the rate. How do you determine?
Oh, okay.
You assume from references.
That's a bigger question. Yeah.
Yeah.
That's another
topic.
Yeah, there's a lot of research area. You know, there's the Bayesian inference methods.
Yeah.
So that means if you have some observation, if you have some mathematical formula, differential equation or SDE or chemical reactions or something like that, how to estimate the model parameters to match the experiment data.
Yes.
Yeah, that's another question. So
that's another topic.
That's another topic. If you so some I think in our school we have a unit to discuss this topic.
Yeah. Okay. So again, any questions from the audience? You can raise your hand and ask directly. I think it's quite a difficult topic for the students, but it's good they have some insights on. It's like a multidiscipline because there's differential equations, there's statistics, there numerical methods include. So by the way, we have young lecturers here. any possibility of doing like a PhD or master's degree in your department?
Yes, welcome. Yeah.
Welcome. Okay,
Yeah.
just send a proposal to you. Okay.
Send me a proposal, and then I will ask our school to. Yeah, I'm happy to help.
Yeah.
To have
Okay. Okay. That's cool.
together to study together. Yeah.
Yes, we have some in applied mathematics interest.
Okay. Yeah.
Yeah. Okay. No questions from the audience? Well, thank you very much, Tian Hatian. It's good to have you here. We hope we can like continue with more lectures or collaboration in different forms. Okay, I return the screen to Ms. Mega, SDMC.
Okay, thank you very much, Mrs. Ratna, for guiding us through the discussion session. And also thank you to our speaker, Associate Professor Tian Haitian, for a very insightful presentation and a wonderful discussion, although it's very new for us and quite difficult to understand, but it's very enjoying. Okay, now we were going to our next agenda, which is giving certificate, and also thank you note to our honorable speaker, Mr. Associate Professor Tian Haitian, by representative of Mathematics Department, Mrs. Ratna Heriana, MSc PhD.
Okay, thank you.
Yeah, please, Dr. Tian, this is the certificate of appreciation, token of appreciation of you delivering the lecture in our department. Hope we can have another session with you in the future.
Okay.
We will send you the e-certificate with the signature later once we get the signature of the dean. Yeah, this is just the ceremonial. Okay.
Okay, thank you so much, Mrs. Ratna, for delivering thank you note and certificate. Yeah, ladies and gentlemen, finally we are going to our last agenda, which is closing. Let's close our program by praying together. Pray, begin. Finish. Okay, I would like to say thank you for all guests and participants for attending today's visiting lecture, and thank you for your active participation and also attention during our event. I am your Master of Ceremony, representing the entire committee. Apologize for any mistreatment present at this event. Thank you again. See you at next visiting lecture.
Thank you, Dr. Tian. Thank you. Let's give applause to Dr. Tian.
Thank you, Dr. Kelas saya, Assalamualaikum warahmatullahi wabarakatuh.
Thanks. Bye-bye.
Bye-bye. Oh, you left? Okay. Thank you, Ms. Mega. I can leave.
Thank you.
I have this ada slide-nya. Nanti saya share ya kalau ada yang interest.
Susah ya, Ibu?
Ya,